My name is Scott Terhune, and I am an assistant professor in microbiology and molecular genetics. And I'm also in the Biotechnology and Bioengineering Center. Because the BBC is interdisciplinary, we all come from different backgrounds. We all come from different environments and different methods of training. We're all quite unique within this environment, and that allows us to help each other out. So there's a lot of information that I've learned in regard to cell biology and viral infection, as well as molecular biology and to some degree cancer biology. But there's also people with backgrounds in more physiology and systems that are more relevant to human anatomy. So because we have such diverse backgrounds, when we get together and discuss our work, we complement each other's technical skills and knowledge base. And that has really allowed us to think beyond just how a virus and a cell interact. It allows us to think about how does that interaction occur in the context of a system. It allows us to think about much more complex situations that my training alone wouldn't be sufficient for me to pursue those. My laboratory is focused on identifying how viruses manipulate host cells. So that means we used advanced technologies to try to figure out the specific targets the virus utilizes in order to manipulate the environment so it can replicate. So why do we do that? We do that because in order to figure out a cure or a way to preventing virus replication, we first have to figure out how it works. So my laboratory focuses on how herpes viruses manipulate the cells that they infect. And the reason they do that is because the cell has innate mechanisms to block viral infection. And it's the virus's goal to replicate. So the virus needs to stop those cellular mechanisms in order for it to replicate itself, in order for the virus to replicate itself. So we study various aspects very early on in the context of infection of how the virus controls those responses the cell's trying to initiate to stop the infection. And we used advanced technologies to do this. So we predominantly focus on proteomics. And that allows us to go in and look at what viral proteins interact with components within the cell through an unbiased approach, meaning we don't rely on information in the literature. We can go in and do discovery-based science on critical viral proteins and figure out who they interact with. And that information allows us to formulate a very solid hypothesis and go on to define how the virus is manipulating the cell. An example includes recently we've undertaken a project looking at antiviral resistance. And one of the proteins involved is a herpes virus protein, and this is antiviral resistance of herpes viruses. We figured out who the target of that viral protein interacts with in the context of infection, and that has allowed us to come up with a hypothesis why antiviral resistance arises during infection and treatment with the current antiviral drugs. So that's one of the ways we utilize advanced technologies to accomplish our goals of understanding host pathogen interactions.